So if everybody will go ahead and mute yourself until you are the speaker, and then we'll be live in just a moment, I believe. All right. Thank you guys so much for being here today. It is the February 2025 edition of Lunch a la Carte. The CART Fund is pleased to provide this service every month, featuring one of our previous researchers and these folks are doing amazing things around the country. And we're so grateful not only for the work that they do, but the time that they take to share that work and their efforts with us. And so just as a reminder, the schedule for these events is on our website calendar at cartfund.org and also on the DAC-DB calendar. If you are a Rotarian, you can find it by selecting multi-district events, and then you can register there. But don't forget, we always have a full library of these videos on our YouTube channel as well, so you can go back and watch any of the previous episodes that you would like. We do have lots of other events coming up in the next several months, so please make sure to keep an eye on our calendar for that. And with that, I'm going to turn it over to one of our past board presidents, Bill Parker, who's going to introduce today's speaker.
Thank you very much, Tiffany. And Dr. Ikezu, we are delighted to have you today. I've observed that most of the research and the drugs for Alzheimer's in the recent past have focused on clearance of the amyloid beta plaque buildup, but very little research appears to be effective in eliminating the tau tangles. So we're excited today to hear your input. And with that, I'd like to introduce Dr. Sania Ikezu, is the professor of the Department of Neuroscience and director of Molecular Neurotherapeutics Laboratory at the Mayo Clinic in Florida. Dr. Ikezu is also associate director of Regenerative Science Graduate Program, Mayo Clinic Graduate Studies for Biomedical Science. Research in Dr. Ikezu's lab is particularly focused on how the innate immune-related cells, extracellular vehicles, and molecules in the central nervous system influence the pathology and progression of select neurodegenerative disorders such as Alzheimer's disease and related disorders. His laboratory was funded by multiple NIH R1 grants, the RF1 grants, and also he received 2 grants from the Cure Alzheimer's Fund, which is an associate member of the Alzheimer's Dementia Rotary Action Group. Prior to this, Dr. Ikezu was professor of pharmacology and neurology at Boston University School of Medicine and led the laboratory of molecular neurotherapeutics for 10 years. He has authored more than 120 journals, which were cited over 23,000 times. By the way, he's also noted as a Google Scholar, so well done on that also. He's also edited the textbook Neuroimmune Pharmacology and served on several editorial boards. Over his career, Dr. Ikezu has received the Vada Kinman Oldfield Alzheimer's Research Award in 2000, the Inge Grunke Equal Award from Alzheimer's Association in 2016, and recently the Jack Spivak Excellence in Neuroscience Award in 2018. Dr. Ikezu is co-chair of the Extracellular Vesicles in Nervous Systems, or the EVINS, Special Interest Group, which is sponsored by the International Society of Extracellular Vesicles for its inaugural symposia. Dr. Ikezu is also a regular commentator of ALZ Forum, which is a forum for discussing Alzheimer's disease causes and possible treatments. Dr. Ikezu received his medical degree and his PhD from the University of Tokyo School of Medicine and has completed postdoctoral training at Massachusetts General Hospital and the Cleveland Clinic Foundation. with that, I turn the program over to Dr. Ikezu, and welcome. I'm sure glad we get to hear from you.
Thank you, William, for very a generous introduction. And it's my pleasure to introduce updates on my research progress since I moved to Mayo Clinic in Florida in 2021. And then I actually visited Colombia to meet with CART Fund members a few years ago for the second award presentation as well. So nice to meet you again. So let me share my screen and then start my presentation. Okay, can you see my screen? Good. Okay. So today for this CART Fund Luncheon seminar, I'm going to talk about the extracellular vesicles and microglia, the therapeutic target of Alzheimer's disease and related disorders. So I received the first CART Fund in 2014. At that time, the research title was Identification of the Mechanisms that Spreads Tau via Exosomes in Mouse Brain. So in this proposal, we mentioned, let me change this to monitors. Okay, so we proposed that this dying neuron containing toxic tau protein can secrete tau protein to nearby recipient neurons to see how the disease spread. And this is known to be mediated through secretion of the soluble tau protein, but we argue that there's a significant involvement of the microglia, which is the immune cells in the brain, by secretion of extracellular vesicle, which is a small particle, which is readily uptaken by the nearby neurons. And this paper was published in Nature Neuroscience in 2015 after this funding. As William mentioned, I also received the Inge Gryke Iqbal Award, which recognized the most impactful paper in the last 2 calendar years from the Alzheimer Association International Conference in Toronto in 2016. Also received several NIH awards to continue this research. So thank you very much. So the brief introduction of Alzheimer's disease, this is the most prevalent age-related dementia. So that when people age, you lose the synaptic connections in the brain, and Alzheimer's disease really accelerates this process and leading to the early onset of the dementia before the end of the lifespan of the human. And when you look at the brain pathology of the autopsy brain of these cases, you can see representative pathology. So this was the Vyshorski silver staining of the Alzheimer's disease patient's brain. You can see a hallmark of pathology called plaques here, and also the tangles, which is mostly consistent with the aggregation of tau protein neurons. And in this plaque, when you look at the surrounding cell types, you can see that this plaque is surrounded by the activated astrocyte or astroglia. This is the glial cells. This is the second major population in addition to neurons in the brain. And then also the microglia. This is innate immune cells, similar to macrophage or monocytes in periphery, which play a role for clearance or compaction of the amyloid plaque in the brain. So the pathology of the Alzheimer's disease is complicated, but different pathology process, different cell types. So what we wanted to know is which part of the pathology or cell type is driving this disease to one place to another. So when you look at the time course of the Alzheimer's disease, it's known that this is from the adapted with the Sparling's Alzheimer's dementia paper. You can see the accumulation of the amyloid beta peptide, which is a major constituent of the senile plaque in the brain before the onset of the Alzheimer's disease. So this red line shows the memory loss. So you can see the A beta accumulation way before the start of the memory loss. And then after the A beta accumulation in the brain, which you can detect by the PET imaging, you can start seeing the synaptic loss and also tau accumulation in the brain. And when you look at this memory loss and tau accumulation, you can see the cross-correlation documented tau protein in the brain with memory loss. So actually, tau accumulation is more closely correlated with the loss of memory in the case of Alzheimer's disease brain. So right now, the amyloid therapy is targeting this A-beta accumulation. But usually, by the time you start seeing the memory loss, most of the A-beta accumulation completed in the brain. So it's very difficult to do early intervention of the A-beta treatment unless you do A-beta PET imaging or biomarker screening to stop this accumulation before the onset of the disease. So the second option would be the targeting tau protein, and which, as William mentioned, we are still behind. We don't have FDA approved drug. So this is a space that we have to put the effort for the second generation drug development for the Alzheimer's disease. So how this tau pathology develop in the brain. So this is from the Brock and Brock. This is a very established neuropathologist. He make a Brock staging or summary from more than 1,500 cases neuropathologically characterized. The stage first start from the tau accumulation in transcendental region or renal sulcus. So this is the stadium tau with 88 antibody, which recognize the phosphorylated tau protein. So this aggregate protein is phosphorylated. This 88 is very good for detection of the misfolded hyperphosphated tau protein in the brain. And then stage 2, you can see the tau accumulation into the internal region or layered internal region to the CA1, CA2 region of the hippocampus. So this is called what we call silent stage of prodromal AD stage, because this can be seen in a normal aged population as well, called the primary age-related tauopathies. But with the pathology spread beyond the hippocampus into allocortex or occipitotemporal gyrus, then it's what we call limbic stage or mild cognitive impairment stage. You can start seeing the sign of cognitive dysfunction. So if you can detect this tau pathology development within the internal hippocampus, then there's a window that you can prevent the onset of the early cognitive impairment. And how this tau protein is consistent with. So this has a lot, several components in the tau structure. I don't get into the details, but the important point is this microtubule binding domain. This is 3DP, 4DP. This part is play a role for the aggregation. Coincidentally, the proximal region of this tau protein is also hyperphosphorylated. So this phosphorylation seems to play a role for this fibril formation. So there's a recent discovery or development of the technology for the crystal imaging of the tau proteins by cryo-EM. So in this case, instead of crystallization, you can image the several kinds of tau structure in the frozen conditions. And from here, this is one of the representative structure. So this is like a C-shape. So this filament consists of C-shaped structure, and then this structure represents this microtubule binding, so which play a role for this data sheet helical filament in the brain. So usually tau is non-structural protein binds to the microtubules to sustain the axonal structure, but once it's misfolded, it dissociates from micro axons. And then it induced the destabilization axon, induced degenerations. And this tau oligomers can spread from neuron to neurons. When you look at the anatomical structure, you can see internal cortex, hippocampus to cortical region. These are basically based on the projection of the axons from the neurons. So the tau can migrate into the axonal terminals and then seeded into recipient cells, in this case to the postsynapses. This can be secreted as a soluble form or the vesicular form. And then our laboratory and others are studying that this microglia activation astrocyte also play a role for this spread. How it is involved, which is our main theme of our research. And we are focusing on mainly microglia and also extracellular vesicles for this tau protein spread from cell to cells. And then, so the extracellular vesicle, these are very, very small particle of nanosize. It can be anywhere from 50 to 150 nanometer in the conventional exosomes. And then the larger one can be anywhere from 100 nanometer to 1 micrometer. So this is the size of the viral particles. They're much smaller than bacteria. And it's released from any type of cells. And this contains many of the cellular components because it's originated from cells. So it has protein, lipid, nucleic acid, and these are basically disposed from the cells. And many people thought this is basically a disposal system from the cells to remove the wasted materials. But actually, these also contain the aggregated proteins, misfolded proteins, and then some of the RNA, such as microRNA, play a role for the messengers to transmit information from cell to cells. So in the central nervous system, different cell type, such as neurons, microglia, oligodendrocytes, and also astrocytes, can communicate information through secretion or reception of these extracellular vesicles. So how these EVs play a role for neuron-glia communication is a hot topic in terms of neuroscience. And this has application to understand how the pathology is spread by delivery of the misfolded protein along with some of the key molecules inducing neurotoxicity, microglia activation, astrocytic activation, oligodendrocyte dysfunction, and so on. So we actually isolated extracellular vesicle, which is done by Satoshi Muraoka when he was in my laboratory. We isolated this extracellular vesicle from APP mouse brain, which developed amyloid plaque in the brain. And then we did protein profiling to understand what's the difference in the protein composition between the APP mouse and the wild-type control mouse. And after protein profiling, what we found is that there's accumulation of disease-associated proteins such as APP or presenilin. These are causative of the autosomal dominant Alzheimer's disease along with the ITGAX, which is a marker for the activated microglia. So we found that there's a cross correlation of the ITGAX ApoE in microglia gene expression. It's the enrichment in extracellular vesicle in the brain. This suggests that in the context of the Alzheimer's disease, there's amyloid plaque formation. And this plaque associated microglia, which is shown in the blue, is secreting this extracellular. So this is a kind of illustration from this publication that was that made a cover page of this journal, and then they kindly made a very nice illustration to summarize this finding. So the take-home message from here is that the plaque that we see in early stage Alzheimer's disease brain accumulate microglia into that plaque region, leading to the secretion of the extracellular vesicles. And this is a scheme that this cryo-EM technology is not only applicable to protein structure, but also high-resolution pathological imaging as well. So if you apply this cryo-EM technology to the IPP NLGF knock-in mouse model, this is the Alzheimer's disease mouse model, you can see this filamental structure of the plaque. So this kind of plaque aggregation in the scheme is actually like this in the real structure. It's a filamental structure of the fibrils. And you can see this structure is decorated with many vesicles. So these fluorescent colored materials are extracellular vesicle, which is imaged by the cryo-electron microscopic imaging. So when you compare the APP-NGF mouse, this is the amyloid mouse model compared to wild-type mouse. You can see about 100-fold accumulation of this extracellular vesicle structure to the plaque region compared to wild-type. So there's a lot of plaque extracellular deposition to the plaque in the mouse model. And we argue that these glial cells, including microglia, may play a role for this extracellular secretion, which seems to be accumulated in this plaque region. And this is another paper from Gilbert's group in the human AD brain. They did similar approach, cryo-image structure analysis of the amyloid plaque. And then here, this cyan image is the filamentous of the plaque, which you can see in APP mouse too. And then you can also see the decoration of this plaque by the organella, including the extracellular vesicle. So it's very similar. Structure can be seen not only in animal models, but also human brain. And we isolated this extracellular vesicle human brain and see whether it contains tau protein or not. And answer is yes. This is a cryo-electron microscopy. So this is a single extracellular vesicle. This dot is detecting the phosphorylated tau protein by the antibody called the PHF1. And then when you look at this, when you isolate tau accumulate fibril or tau aggregates from the extracellular vesicle and did similar immunostaining, you can see this control tau fibril has what we call paired hierarchical filament, PHF, decorated to the same tau antibody detected by the gold particle. Tau oligomer is like the globular structure. So the tau in extracellular vesicle is a globular structure. So it's not fibril, but it's more like a fragmented fibril, which is in this extracellular vesicle that you isolate from Alzheimer's disease brain tissues. Then recent paper from Dan Falcon's laboratory published in Nature Neuroscience last year. This is another cryo-image structure of the now extracellular vesicle. I presented the cryo-image structure of the filament and the pathology, brain section around the plaque region. Now this is the extracellular vesicle cryo-EM. Surprising or astonishingly, they identified this paired hierarchical filament, which I presented here in the extracellular vesicles here. So these yellow dotted ones are the literally paired helical structure in the brain. So demonstrating that not only immunoelectron microscopy, but the real structure, there's a paired helical filament in the brain. So when you inject these extracellular vesicles or from human brain, or the tau fibrils or tau oligomer of the same dose, in this case, we inject a 300 picogram of tau in an EB form, oligomer form, or fibril form into this hippocampal region of the mouse brain, aged mouse brain. You can see the spread of the tau accumulation. This is the recipient cells into this, what we call dentate gyrus, which is the hippocampal region. When after injection of the extracellular vesicle for tau protein, but the same amount of tau in either oligomer form or fibril form doesn't have much tau accumulation in the recipient neurons. So this shows that the EB tau is more efficiently spread in the tau pathology compared to the oligomer or fibril form in the same amount. So we discussed about which form of the tau is efficient in transmission of the tau pathology from neuron to neurons in ARIA scheme. We propose that EB form is more efficient in spread of tau pathology. So this is the current working hypothesis that this entire cortical neuron spread the phagocytosed by microglia, and then this is uptaken by the recipient cells. So that is, as I mentioned, published already. So the journal kindly created this animation, which is also available from the YouTube. Now, moving forward, so this we did it in a wild-type mouse, aged wild-type mouse, and we wanted to know whether it can be accelerated in the APP mouse brain because this contains plaque and activated microglia. This APP mouse has more secretion extracellular vesicle. So this ED-mediated tau propagation may be accelerated in the previous line of plaque, which also gives us the understanding that why the amyloid deposition can accelerate tau pathology in the human brain as well. And then, so this is one of the image. Let's see. So here, this is the microglia expressing the CD9 emerald green. So this is a way to label the microglia derived extracellular vesicles in the human in the mouse brain. So this magenta color is the phosphate tau protein. So in this mouse brain, you can actually quantify how much extracellular vesicle is secreted from single microglia, because you can quantify individual EVs using this confocal microscopic imaging with an enhanced resolution. And so this is a snapshot of what I just presented. And then using technology, you can clearly show that this plaque-associated microglia, which is as a MAC2 positive, has more EV secretion compared to the non-plaque-associated microglia. So the scheme I showed in a post-cover letter in the journal is actually confirmed by this technology. So the current mechanism, so conventional idea is that tau spread from neuron to neuron by secretion the misfolded tau protein as a soluble form. But in the case of plaque deposition in the brain, this amyloid plaque inactivates synapses. And then this inactive synapses usually show eat-me signals such as binding or complement. So this usually efficient phagocytosed by the microglia. So microglia phagocytose these inactive synapses. In this process, they also uptake misfolded tau protein in the synapses. And if the microglia is very healthy, they can do intercellular clearance of tau protein. But if it's microglia is not efficient tau clearance due to the aging, oxidative stress, or genetic predisposition, such as ApoE4, then the microglia spit out the undigested, especially aggregated tau protein into the outside, some as a soluble form or extracellular vesicle form, and this can be uptaken by neurons. So I think in case of Alzheimer's disease, this process seems to be activated due to the deposition, plaque deposition, and that seems to accelerate the tau pathology in the brain. So next question is what molecule can be targeted to suppress this the microglial extracellular vesicle secretion? So we did some kind of molecular screening in vitro system. So first, we used the microglial cell line, and then we transfected the EB reporter molecule. So in this case, CD63 is known to be the marker for the extracellular vesicle. So if you put the red fluorescence, we call it TD tomato because of tomato color, you can see the CD63 EB as a red fluorescence in the cells. So this is one of the fluorescent image of the DB2 microglial cell line. Transform this CD63 TD tomato expression. And interestingly, when you treat this cell line with a high concentration of the adenosine triphosphate or ATP, it rapidly secretes extracellular vesicle into the extracellular domain. So you can see this is a before, right after the ATP stimulation. Within 15 minutes, most of the EB is secreted from microglia. So this is a good platform to screen which gene play a role for the secretion of the EB. So in this case, if you can target gene which control the EB secretion from microglia, ATP stimulation doesn't induce the TD tomato positive EB secretion. The signal stays there. So you can separate the TD tomato positive cells, which is resistant, and the TD positive negative cells, which is sensitive to ATP stimulation for EB secretion. So we use the short hairpin RNA system. So this is shRNA is synthesized from the nucleus. And then after this processing, this shRNA processed RNAi, RNAi can bind to the target sequence, either messenger RNA for cleavage or the transcription silence. So basically, shRNA is designed to do screening of gene silencing. So in our case, this part, TD tomato, so we have a red cells containing this CDC TD tomato. We put a library containing different kind of shRNA targeting at least this more than 20,000 target genes. And this contains the genetic fluorescent protein called DAP-GFP. So after this library construction, the cells contain red fluorescent for the TD tomato and then GFP for the shRNA. And we screen a lot of cells for this. The first input is 138 million cells because we have to use many libraries. And then we screened about 177,000 unique shRNA from this project, which is done by Dr. Lee 01. And then we finished it. It is published in the Cell Report about 3 years ago. So here, this is the CD60 positive cells, TARB-ID positive cells, TARB-GFP positive cells. So we collected top 1%. This is resistant cells due to the gene silencing. We also collected the bottom 1% of TD positive. This is still sensitive to the ETP stimulation because they depleted TD tomato signal after the stimulation. So we collected these cells, did transcriptomic analysis, and then determine which shRNA is enriched in this resistant population compared to the sensitive population. Top 1% side against bottom 1%. And then here, this is the result. After the D-scoring, you can identify the top 10. And these are the lists which we already published. And after some validations, the take-home message that we identified 3 molecules which strongly make this microglia resistant to EB secretion. One is the SEP1, MCFD2, and the CSDC1. I think this is just the alphabet, but each protein has a function. And then from here, we wanted to know which molecule is a good target for suppressing microglial EB secretion. And for there, we focused on the SEP1. It's a selenoprotein 1. This is a soluble protein, not to be secreted mostly in the liver, in the tissue, but in the brain, microglia seems to be one of the source for this SEP1 secretion. So this is another cell report paper we published last year. What we did is we specifically target SEP1 with individual shRNA. And then we make sure that the SEP1 is silenced in this microglia. And then we did more detailed analysis what will happen after this silencing by nanoimager. This is a super resolution microscopy. And the flow nanoanalyzer. This is a analysis of the extracellular vesicle in a single vesicle resolution. So these are high-tech, ultra-sensitive technology for EB characterization. So this is one of the data show that when you target SEP1 with 3 different shRNA clones, you can see the reduction of the EB secretion from the cells. So this is highly reproducible. So this is interesting data. So this is the D-storm. So basically a super resolution microscopy. When you zoom in each puncta, each puncta represent the one extracellular vesicle, which is like 100 to 200 nanometer in size. And here you can see the dots. Dots represent each molecule of the SEP1 on the EB. So we are looking at single molecules. How many single molecules? SEP1 molecules on the extracellular vesicle on one single vesicle resolution. So here you Dr. Victor Santos quantified these numbers. And we show that the loading of the CD60TD tomato, which is a marker for the extracellular vesicle, is reduced by the silencing of the SEP1 in the microglia. So this is another, let's see, data show that, I think I lost my, yeah, here, sorry. This one. So these are different type of the tracker. So in this case, instead of TD tomato with green frozen protein, and then these individual puncta show extracellular vesicle in this BB2 microglia cell line. So it's a little difficult, but if you see the detail in detail, you can see the flow of this EB coming from the cellosoma to the process. So microsynthetic process through the end of the process in this study. And then we injected this into this mouse brain to understand how SEP1 silencing can change the EB secretion from microglia, as we did before. And this is one of the picture. I think it's, maybe you can start from here. It's very similar to what I showed before. And it's kind of sluggish. Yeah, so basically what he did is he quantified the vesicle coming from single microglia after the infection of this reporter gene in the mouse brain, like this. Then he showed that we should do shRNA-based silencing of SEP1 in microglia in vivo, not in vitro, in the mouse brain. You can see suppression of the extracellular vesicle secretion from microglia in this study. So we argue that this EB secretion is regulated by the SEP1, and there's a more functional studies there, but what we found is that there's a silencing of the EB biogenesis part, and the more increased level of the RAMP1, which enhance the lysosomal degradation. So this SEP1 silencing is switching this the intercellular protein metabolism from EB secretion to intercellular degradation of the proteins through lysosomal pathways. So that is was interesting study to understand how you can enhance the degradation of the proteins instead of secretion by targeting the SEP1 in the microglia. So this is one of the target to control the microglia and extracellular vesicle mediated spread of the pathology in the mouse model and potentially human studies. And then I'd like to touch base of the application of this EB in a biomarker as a separate topic, because I think it's really important. So because the reason for this is that since these EB contain many misfolded proteins, microRNAs, and other cell type specific molecules, we can get more detailed information of what kind of change you can see in a disease condition in the different cell types by collecting the cell type specific extracellular vesicles in the peripheral sources such as blood, plasma, or cerebrospinal fluid. And this can be applicable for different early detection or differential diagnosis of the neurological disorders such as Alzheimer's disease, amyotrophic lateral sclerosis, frontotemporal lobar degeneration, or progressive nuclear palsy. So this is the paper from German group, Anja Schneider's laboratory, published in Nature Medicine last year. They actually collected extracellular vesicle from plasma, and then they quantified the amount of the 3-repeat, 4-repeat tau. And interestingly, when you check the ratio of the 3-repeat, 4-repeat tau, it's highly high in behavioral variant frontotemporal dementia, which is known to have a TDP43 pathology in the brain. So this can be a really distinguishable marker between this particular disease from the other disease phenotypes. And when you look at the TDP43 level extracellular vesicle they collect from blood, it's highly elevated in ALS cases. or behavior FTD, because these two diseases has TDP-43 accumulation in the brain compared to the progressive supranuclear palsy or control groups. So this demonstrates that the EB can be utilized for this kind of differential diagnosis of other major neurological disorders in addition to the Alzheimer's disease. So what kind of cell type marker can be utilized? So these are all reported marker for different cell types that can be detected in ectocellular vesicle. So these are candidates to enrich the cell type specific EB marker from the peripheries. And our laboratory has been focusing on the ATP1E3 as the reproducible marker for neuronal ectocellular vesicle that you can detect in the blood as well as CSF. So this one is coming from our unbiased screening of the ectocellular vesicles from the iPSC-derived neurons, astrocytes, oligodendrocyte, and microglia. So this was published in Journal Ectocellular Vesicles about 3 years ago. And we collected EB from the in vitro differentiated neuronal cells and they did protein profiling. And from here, we identified the ATPN3 is a good marker for neuronal EBs, which we published in the Science Advances 2 years ago. And so this is one of the data from this paper showing that you can detect ATPN3 positive and also CD9, which is the EB marker positive EBs in the iPSC-derived neurons, brain-derived EBs, and also CSF-derived EBs, as well as a plasma. So we did a head-to-head comparison of the ATPN3 at the other known neuronal marker called L1CAM. And we found that we tested all 4 different samples, neuron-derived EBs, brain-derived EBs, CSF-derived EBs, plasma EBs. Basically, ATPN3 positive EBs is more detectable than the L1CAM positive EBs. For example, neuron-derived EBs, 20% EBs are ATPS positive, but 0.1% is L1CAM positive. In plasma, 8.1% of total EBs are positive for ATPS3, but only 0.2% of the total EBs L1CAM positive. So I think ATPS3 is more abundant than the L1CAM in all these 4 different samples. And more interestingly, when you combine ATPS3 with A-beta, you can see the more frequent detection of these double positive EBs in the plasma in the AD cases compared to the mild cognitive impairment cases or control cases. The effect size is strong. It's about 6-fold compared to the control or MCI. So effect size is really important to make a threshold for the diagnostic. Interestingly, this ATPS positivity has a positive correlation with the Braak state of the patient, negative correlation with a mini mental status examination, and also negative correlation dementia, dementia rating scores. Yeah. So this can potentially utilize for monitoring the pathology development as well. And then we did the accuracy and sensitivity analysis in using the plasma EB. So we found that the area under the curve is about 0.95 when you use the combined ATPS3 and A-beta compared to the control. So you have very good distinction of the control against AD using these markers. We also see the good separation of AD against MCI cases with area under the curve of 0.54. So this can give you the potential of the ATPIC positive EB as a good diagnostic biomarker for the detection of the Alzheimer's disease cases. So we try to continue this research in the context of biomarker study as well. So summary, we discussed that there's a neuron-derived ectocellular vesicle in the brain, which can be secreted in the bloodstream, and which can be detected as ATPase positive EVs. And then using this single EV detection system by D-Storm, you can see the more enrichment of the A-beta ATPase positive EVs in AD cases compared to the control cases. So that's all of this presentation. I'd like to really thank all of our laboratory members and collaborators, especially the underlined members, Yang Yu, she's a professor in my laboratory. Seiko, she's another assistant professor and assistant consultant too in my laboratory, and Linda Zhang. Victor published a recent paper and cell report. So these are the authors of the paper that I presented today, along with former postdoctoral fellow Zi Wang and Satoshi Muraoka, and also, sorry, Sean Kiron, Kevin Creighton. They also participate in the animal studies and proteomic study that I presented today as well. These are the funding sources. I'm also the part of the, as William mentioned, ESTOS EB in nervous system special interest group, which so the ICF is promoting this EB research in neuroscience. I'm part of it. And this is my the members, and this is my lab space in beautiful Jacksonville, Florida. Thank you very much.
Thank you. Bill Parker, do you have
I'm glad you
something?
simplified this. I can understand now why some of the major pharmacological groups have backed out of trying to come up with the ultimate Alzheimer preventive pill. This is incredibly complicated. And as your tools for analysis get more specific, you discover that it's even more complicated than anyone could have imagined when starting the study. That was incredible. Thank you very much.
Thank you.
Thank you, Bill. It is very complicated for those of us who have no scientific background at all. So I do appreciate you trying to make it more easier for us to understand. Do any of you guys have questions for him while we have him here? Greg, go ahead.
Well, thank you very much for meeting with us this afternoon. I just wanted to make a simple observation. How important was CART when you were starting some of this research? How important was receiving a CART grant?
Oh, it was extremely important because at that time I didn't have NIH funding. So I rely on non-governmental foundations such as Alzheimer Association, CART Fund. So that was really helpful. And especially, I was a junior, mid-career researcher, and then I was actually struggling. So, and also there are many junior investigators who has to get started. So this CART funding is really beneficial for young investigators.
Well, that helps us kind of help prioritize for our Rotary members and others how important it is to continue to support the CART mission. I really appreciate your time, and thank you, Tiffany.
Anybody else? We had a couple of new folks that joined us today, some that came in and out. I'm sure they will catch up on the recording as well. This has been great. Thank you so much. I know that it's been a few years since you had your grant, and so you'll continue to work on this project, I know. And anything else that you want to share with us about what's going on in your world down in Jacksonville?
We are very interested in nucleic acid therapy as well. So that's a next generation initiative that our institution is putting a lot of effort. And we're also putting effort on extracellular physically mediated drug delivery, nucleic acid delivery. So this EB also has the as a technical application for drug delivery as well. So this is another effort we are putting here as well. So diagnostic therapeutics, drug delivery development are the three major emphasis that we are working on.
Wonderful. Well, thank you so much for spending time with us today. We hope that we get to see you when we're in Jacksonville in a few months. And we certainly will be in touch. And we appreciate you taking the time today to spend some time with our folks answering our questions.